Protein characterization. In this last section, we are going to explain some of the most usual characterization methods carried out over the purified samples, such as peptide mass fingerprinting, microdialysis, analytic size exclusion chromatography, circular dichroism, dynamic light scattering, and protein activity. Peptide mass fingerprinting. Even though the purified protein has the molecular weight of the protein of interest and a histidine tag, just in case an identification of the protein is performed through the peptide mass fingerprinting technique. To this purpose, the final sample is loaded in acrylamide gel, later stained with a comazine stain. The stained band or bands are cut from the gel and kept inside a tube that can be frozen at -20°C until the moment of the analysis. The protein contained in the band is cleaved by specific proteases given to small peptides whose masses are determined by a mass spectrometer such as MALDI-TOF. A mass spectrum is obtained where each peak represents a molecular weight of a particular fragment of the protein. For unknown proteins, the peptide fragments are compared with a sequence of proteins from databases. However, in no case, the peptides are compared to the known sequence of the produced recombinant protein. The results of the comparison are statistically analyzed given a histogram. A list with possible matches is given. The best score will represent the identity of the protein as it can be seen in this table. So in this way, we can be sure that the protein obtained is the desired one. Microdialysis.
The objective of this method is to screen different dialysis buffers once your protein is purified. The idea is to put in contact a small part of your sample with the buffers through a filter membrane to easily see if your protein precipitates. Then you can choose the buffer in which your protein is soluble.
In this experiment, we are going to microdialyze two different proteins with those filters, and we have set 3 conditions per protein. So we have 3 different buffers, and we pour 20 milliliters of buffer in each of these Petri dishes. So we take individual filters and we put on the surface of the liquid, and after that we put some more others. You can add more than one in each of the plates. For each filter you can microdialyze around 60 microliters in drops of 10 microliters each. So what you have to do then is to set the pipette to 10 microliters and then dispense in this on the filters the 10 microliters sample in individual drops. And you can load more than 1 drop per filter. If you separate conveniently, you can even load around 6 drops or 7 on each of the filters. So depending on the final volume of protein that you need for the experiments that you have to perform afterwards, then you may have to calculate the number of filters and number of drops. Then you set the plates at room temperature during 30 minutes, and after that time you take the different drops from the filters and be careful to avoid the interaction between the buffer and the protein drops, because then you will have dilution of the sample and you are not going to be able to analyze what has been the efficiency of the process in the individual dish plates. Okay, so once you have all the contents in a single Epender tube, you can then analyze the result. In order to analyze the solubility of each protein sample in the different buffers tested in the microdialysis experiment, it's necessary to centrifuge all the protein samples, and this is done in a refrigerated microcentrifuge for 40 minutes at 15,000 Gs. After the centrifugation steps, all the supernatant of the protein samples will be transferred to a new Epender tube to eliminate the pellets which contain the protein that has been aggregated during the process. Finally, the amount of protein that remains in the supernatant can be measured in an spectrophotometer at 280 nanometers, provided that the signal of each of the buffers is subtracted prior to the reading of the protein samples. And with that result, you can take into consideration the initial amount of protein that you loaded on the filters and the final soluble protein that you obtain.
Analytical size exclusion chromatography. The analytical SEC is not a purifying method, but rather is used to look at the protein size profile, homogeneity, and aggregation. This step can be necessary, for example, when proteins have been stored for a long time or even frozen. In this case, because a small aliquot of the total sample is going to be analyzed, the small size column Zapardex 200 increase 10-300 is used. The maximum amount of sample allowed in this column is 500 microliters. In this case, a loop of 250 microliters is prepared to introduce the sample that is going to be analyzed. In this case, a sample of purified green fluorescent protein is going to be analyzed. It's introduced in a small syringe and the bubbles are removed to avoid the entrance of air in the FPLC system. The 250 microliters of the protein are introduced inside the small loop. The exit of the green protein ensures that the loop is full of sample. The SEC method used for this particular column is selected and run, checking that all parameters are correct. The sample is injected on the top of the column. The running buffer moves the green protein to the bottom, clearly seen in this green strip. When the sample leaves the column, it's detected by an increase in the ultraviolet absorbance. This chromatogram will indicate that most of the protein is homogeneous and only some aggregation has been produced during the protein storage. Circular dichroism. Circular dichroism is a spectroscopic technique for structural characterization of proteins. The differential protein absorption of the left and right-handed circularly polarized light gives information about their secondary structure. The protein secondary structure is determined by the absorbance of the peptide bonds in the ultraviolet wavelength. As an example, here we show overlapped circular dichroism spectra of membrane proteins. In red, the typical spectrum for proteins having predominantly antiparallel alpha helical bundle. In blue for proteins with predominant beta barrel, and in green for proteins with mixed helical, beta sheet, and unordered structure. Dynamic light scattering. Dynamic light scattering, DLS, is a technique to determine the size distribution profile of small particles in suspension. To this purpose, a laser is shot into a sample and the light is scattered in all directions when it impacts with the particles. The diffracted light is captured by a detector. The scattering patterns are automatically analyzed, giving as a result a sized particle profile. This technique is very useful to determine the diameter of spherical particles, such as protein nanoparticles or liposomes. Protein activity. In some cases, it's necessary to obtain the protein in an active state. In this case, specific activity assays must be performed over the purified proteins. The method must be specifically designed to the type of activity of the protein to be analyzed. Interaction assays can be performed by using, for example, a surface plasmon resonance that allows to analyze the affinity of ligands in several conditions to the purified protein. This is possible by measuring changes in the light refraction shooted upon the absorbed molecules in a functionalized chip. With this last section, we have finished the videos about our principal service activities. We hope you have enjoyed it. These videos have been performed by the Protein Production Platform staff. Dr. Neus Ferrer Miralles, PPP Director, Dr. Merce Márquez Martínez, PPP Technical Coordinator, and Rosa Mendoza Moreno, PPP Laboratory Technical Manager. We want to thank the Nanobiotechnology Group for their support in the elaboration of some of the videos. Thank you very much for your attention.